Hello everyone. In this video of image processing and computer vision, we are going to talk about image segmentation, which is texture based. So if you have an image of some object or some scene or anything that has different textures, we want to segment that image into regions, each one having a separate or distinct texture. And for texture analysis, we're going to use Gabor filters. And for segmentation, we're going to use Kmeans clustering. So since I have never covered Gabor filters in my previous videos, I want to briefly mention that, and then I'm going to show you how to do this segmentation. Now I have videos on Kmeans clustering, so I just need to cover Gabor filters. So a Gabor filter is a linear filter. First of all, that is very important because applying linear filters can be done by convolution, and it is much faster than nonlinear filters like a median filter or something. We use it primarily in texture analysis. And if you look at it, it's basically a Gaussian filter. You see, it's a negative exponential of something squared times what? Times sine and cosine. Now you might say this is another exponential. That's right. But remember, it's exponential of I times something. So, based on Euler formula, this is going to be cosine of something, right? Plus I times sine of something. So it is really a Gaussian filter or wave that is modulated by a sine wave, right? So when you have this sine wave that is going back and forth, right? If you apply a Gaussian wave on the top of that, then what you will have is that sine wave, but limited only to a special region. So let's say this is my Gaussian wave. When you put that there, then this two will get bigger basically weight, these other ones will get a smaller weight, and you basically get something similar to basically something like this. Okay, so a sine wave modulated by Gaussian, or the other way around, the Gaussian modulated by sine wave. So the good thing about it is, first of all, it captures frequencies because you have sine wave and you can change the frequency of the sine wave. Second thing, it's limited to only a special region and it doesn't go all over. So when you basically look at it here, these are how they look like. When you look at it in 2D, right, you see clearly this is a sine wave, which is only going in this direction, right? It has, and you can control the orientation of it, of course. So if I want to measure a sine wave in the X direction, then I set my parameters so it only measures sine wave in the X direction with a specific wavelength. So if you want, you can change the wavelength, or we call the scale. So you see all of these four share the same orientation, x-axis, 0 degrees, but their scale or wavelength is different. This one has a smaller wavelength, and as you go forward, it has a bigger wavelength. So you can get different, basically, frequencies, and of course, you can change the orientation, right? So now you are going to 30 degrees different wavelength, and you can go to 60 degrees and so on. So you can get different types of textures by convolving these kernels, Gabor filter banks, we call it, with your image. So the equation for that, as I said, is a Gaussian times a harmonic, right? And it's a complex thing. So when we look at the Gabor filters, the result of applying or convolving an image with the Gabor filter, we need to typically look at the real part of it or the absolute part of it, okay? Because they are complex numbers. So we have to only look at the real part or the absolute part to see a number that is not complex number. Now you see I have lots of parameters here that are under my control. I have lambda, I have theta, psi, sigma, and gamma. So what do they do? Clearly, if you can look at lambda, lambda here is in the denominator of your sine function. So clearly it controls the wavelength, or in these pictures, it's the scale basically that you're controlling. Theta here, if you look at your theta parameter, that's the orientation of your function. And you might say, well, there is no theta in this formula, right? Where is theta? The thing is, if you look, G is a function of x and y, but here instead of x and y, you have x prime and y prime. So this x prime and y prime are not x and y, they are the rotated version of x and y, and in that rotation you are going to see the angle theta. So what is that? So here I would add it for you. Look, so here x prime and y prime are x cosine theta, y sine theta, and y prime is negative x sine theta and y cosine theta. So they are rotated by angle theta about the z-axis. So clearly here theta is showing your rotation or orientation of the filter. Lambda, as I said, is the wavelength, which is the scale of the filter, right? Then what else we have here? You can see that there is a sigma. Sigma shows the spread of your Gaussian part. So it's basically the standard deviation or the spread, right? So how far does it go? Is it just this much, or it is going to be a little bit bigger or so on? So how local you want to measure that? Then we have this lambda, this gamma here, and gamma here, because if both weights of x prime and y prime squared is 1, that is going to be circular. But when you add this gamma, you are adding an aspect ratio, and so you can make it elliptical. So basically, you can, instead of having this Gaussian to be, if you look at it from the top, to be like a circle, you can make it to be like an ellipse by picking what? Different gamma. If gamma is 1, yes, of course, you get a circle. But if gamma is bigger than or less than 1, you get ellipses in different directions. Finally, there is one extra parameter here, and that is a phase shift psi that you have, a phase offset. And really, it is not doing a ton of thing. It's just moving this. Here you see this white part is exactly in the middle. That's where psi is 0. But if you want, you can bring this white part a little bit off center. So you can shift these bands a little bit left and right by using psi. So it is not as critical as the other ones are. Even aspect ratio is relatively good, but I would say the biggest parameters, and sigma as well, the biggest parameters are clearly lambda for wavelength, and also what? Clearly your orientation theta. So lambda and theta are the most important parameters. Then probably I would say maybe aspect ratio gamma. Okay, so these two are the ones that you change. In MATLAB, there is the command Gabor filter and Gabor filter bank to create. These two are the ones that you change. And again, you might be able to go ahead and change the other parameters too. As I said, gamma, and then probably sigma, and finally psi, but these are not as critical. The ones that are critical are these two. So, and these are different, as I said, these are different filters. The collection of them we call filter bank. So when we want to detect different textures and distinguish between them, we have to apply all of these filters to it, all the bank. We have to convolve the image by all of them, get different responses. And based on the collective responses of each region to these filters, we decide whether that belongs to one group of texture or another group of texture. So here is a sample work that 6 clusters are applied here with Kmeans clustering and Gabor filters with 5 scales and 6 orientations. And why 6 clusters? Because there are 6 types of textures here. So this is one group of texture. The next group of texture is this one, which you only have 2 of that, right? So let me color them for you. Then you have this group here, right? Then you have this group. Then you have this one. And finally, let's pick a different color. Maybe this one, that's interesting color. So a glitter color. Here, that's another one. So you see I have 6 clusters, 6 types of textures. That's why I use 6 clusters. Now, most of the time, if your job is automated, you don't have any idea ahead of time of this hyperparameter number of clusters. So you have to just pick something and try different numbers, see which one gives you the best segmentation. But here you can see that it detected that these two are kind of the same cluster. You can see them, and clearly they are. Look, you see that they are the same. The other ones, it's not as clear. For instance, you can see that this region here and this region and this region, which are these three in yellow, it also kind of detected them as being one, but then the other ones are not as clean. Okay, I mean, if you look, well, kind of, you might say that this one and this one and this one are also the same, which again, these are these green ones. But as you can see, the other three clusters, it does not have a huge distinction. I mean, clearly these two are very different, but you see that they are relatively similar here. So it does not do a great job. And one reason is because you are not bringing in any special information. So you just say, hey, if the textures are different or the textures are similar. Plus, of course, you have these two parameters to pick also. What are the scales? What are the orientations that you pick? And how many of them? That can make a big difference in the result. But here, we are using the same thing, 5 scale, 6 orientations, and I can go with 8 clusters or 6 clusters, but here I'm bringing special information here, which I'll show you. I add X and Y to the texture. So now you have to segment based on X and Y plus whether they are close to each other or not. And now if you look, you clearly see that these two are much better separated than the rest of them. Very clear. You see those dark blue ones. Here you see they are not separated from the rest. Then you have these brown regions. Look at them. This one, and this one, which again, these two, you see. So now these two that were not separated here are separated very beautifully. Then you have these very dark blues here. So you have these two regions, which are clearly these two, very clear. Then you have these red ones, which again, you see they are different regions. Detects them beautifully. Then we have these guys, which are these kind of brick walls, right? So you see it is doing a relatively good job. Look at these three, these cyan colors, right? Look. So you see, it detected them all separately, relatively beautiful. Okay, and here I use more clusters than needed. It will do better than if you go with less clusters than needed, okay? Because it merges some of these clusters together ultimately, and they look very close to each other. But you see that it can do a relatively good job when you bring in and add special information. So these two that are next to each other, they are separated not just because they have different textures, but also because they have different X values. Okay, so that is very critical. And here is the code that I wrote for it, and I'm going to share with you. So in the first part, I'm only doing Kmeans clustering with Gabor, no special information. I read the image, convert it to black and white or grayscale, which should be for the Gabor textures. I use 4 scales and 6 orientations. If you want, you can use 5 scales. Just use any number. And these are the scales that I picked. My orientations go from 0 to 180 with number of orientations plus 1. And then to create the Gabor filter bank, all you need is to just apply the command Gabor and just give it this set of wavelength and the set of orientations. It creates a Gabor filter for you. And here I wanted to show you the Gabor filters. So when you change the wavelength, when you change this lambda, the size of the kernel goes bigger. Okay, and if you just show the entire kernel or the filter, you're gonna see all of them look the same. Why? Because the ones that has a bigger wavelength, it is gonna be bigger size too. So maybe this one is like 64 by 64, this one is like 512 by 512. When you look at both of them, they look very similar. But if you zoom into the middle part of them, that is 64 by 64 for, let's say, all of them, or 32 by 32, in their middle part, that's where you see the different wavelength. So here you see I picked up a crop size in the middle, and I only look at that 32 in the middle of the filter, right? So here I want to show you this Gabor filter. So clearly you see that when you create the Gabor bank, right? First of all, this Gabor bank is not a set of matrices only. It has more data to it. It's a structure. If you look at this Gabor bank, let me show you. It's a structure, right? 1 by 30 Gabor bank, right? And 30 because it's the second part that I use 5 and 6. So you have 30 you see there is wavelength in it, there is orientation in it, there is a special aspect ratio in it, and there is a special frequency bandwidth. So you see there is special aspect ratio, so you can change that gamma if you want to. We didn't change it, but we can. Now the ones that we want, those matrices, are these guys, special kernels. So we have to get the special kernels by dot of Gabor bank I, and then, as I said, these are complex variables, so I have to look at the real part of it. Now that I look at the real part of it, I go ahead and crop the middle part of it out, and then in a separate subplot, I'm going to show you. So if you do that, there we go. Look, here is that initial one with 4 and 6. So you see all the top ones are orientation 0 with different scales. Then you go to orientation 30 all the way to 150. And the reason we didn't go to 180 because it looks the same as 0. So you see these are different wavelength, different orientations, and these are the filters that, of course, these are just the center parts of them. These are the ones I'm going to convolve my image with, different regions of it, and I move them along and get different responses. Okay, so that's the Gabor filter. Now, if I want to apply it to the image, here I have to initialize the Gabor responses, okay? And for that, I start with pre-allocation. So I start with cells. Each cell will include one of those, each component of the cell will include one of the responses. And then so I go from 1 to number of scales, 1 to number of orientations with 2 nested for loops. I'm going to go ahead and I'm going to apply that Gabor filter to that image. And you see the way I do it is by the command Gabor filt. Okay, so you don't need to use convolve, you use Gabor filt, and you apply the Gabor filter here to the grayscale of the image. You get the responses and you collect them, these Gabor magnitudes, of course. And once you get these magnitudes, now you can go ahead and pass them to the K-means. So for the K-means, I have to give it the number of clusters. I go with 6. And then the reshaped feature. Now, what is reshaped feature? When you pass data to K-means, you cannot pass to it data that are in the shape of a simple image. Okay? The data that you have to give to K-means should be 2-dimensional X and Y, but you don't have one X and Y. We don't have one single image. You have a ton of different response images. So what you need to do is you have to reshape all of these different responses that you got. You have to reshape them into 2D. And here we use normalization to get better classification or clustering by normalizing them by their max. So they're all kind of the same range instead of different ranges. And then with K-means, there are 2 parameters that you can give it, major parameters. One is replicates, the other one is number of iterations. Because K-means is an iterative algorithm, if you watch my previous video. So you can tell it how many times to repeat before you say it converges or not. So here I didn't give it, but you can say, hey, 300, 200, 100 iterations. And then replicates, this is the number of tries with different random seeds. So, initially you have to pick a seed, you have to pick a center for each one of the clusters, and you do it randomly. And depending on where you start, you might get a little bit different result. So here I'm asking it to start those seeds 5 different times, right? And if you don't want, you can just say, hey, just do it 1 time or 10 times. The more replicates, the more the number of times the Kmean has to repeat itself and the more time it takes. So, but it might give you better results, right? Kind of more robust results. So once I get my cluster labels, now I can basically uh, go ahead and show my segmented image, okay? And here, so when I ran that, this is the result that I got. And of course here I changed some parameters so I get a little bit different results, but again you see that it detected some of these clusters relatively good, and the ones that are kind of close to each other, it did not do a great job. It kind of assumed that they are kind of the same thing. That's why in the second part I brought in spatial information, X and Y, of the image pixel, and I add one more to my number of scales to capture a better range of frequencies here. So it's very similar again. Everything is the same. Now, you see here, I'm using ABS. You can use real, that's fine. And again, reshaping them into 2D. Now I add the spatial information. So I use mesh grid command, and I go from 1 to the number of columns of the image, 1 to the number of rows of the image. I order them in this format. You see again here I'm vectorizing them so I can convert them to 2D, and then I bring this spatial information, merge it with the Gabor filters, and now I use all of that for my K-means. Okay, again 6 clusters, 3 replicates, and redo everything, and this is the result this time. And if you look at this result, it is significantly better than when you didn't have spatial information. Of course, there are some areas that it doesn't do a good job, like these two textures that are kind of a little bit closer to each other. It kind of considers them like one cluster, but the rest of it, if you look, it very well detected the rest of it. Okay, and here is with 6 clusters, but here you see I did it with 8 clusters and I got better results because I used more clusters. So it forced these two guys that were next to each other to kind of go into different clusters. So if you use more clusters, it's always better than to use less clusters. Okay, and you have to play with number of clusters, the number of scales, and the number of orientations, and then you can get different results. So here you can run each one of these code that you want. You can run the first section, you can run the second section, and get different results. And I'm gonna give you a download link for all of them. So here I'm gonna run it for you with 8, so you can see the result of that before I finish this video. But here is one simple technique based on Gabor and special information to do texture-based image segmentation. Here is with 8 clusters. You see now that these two are very, very well separated from each other. So I recommend using a little bit more clusters. Sometimes you get much better results than using less clusters here. Okay. Now, of course, you might have some anomalies here, right? So it assumes that this portion of this cluster is a little bit separate. But again, overall, you see that if you now on this one, you go ahead and apply some smoothing technique or some median filter and assign the value of that to the entire thing, then you're going to get homogeneous numbers all over and you can get much better results. Okay, so you can further process this result and assign a median value to all of the same region. And by further processing that, you can get much better texture results. Hopefully the video was useful to you, and I'll see you in the next lecture. Thank you.